Guys, we're going to continue our experimental technique. Okay, previously we had discussed about hydrolysis of complex carbohydrates. Okay, because the question asks you from complex. Okay, I'm sorry. First the question. Okay, so first we have settled this thing. Okay, these two things. Okay, the name of process that you use hydrolysis. Okay, so the two types of substance that can be used to break down, we use hydrochloric acid. Okay, the name of a process that used to separate the monomers. Okay, so this is the acid, the enzyme used to break down. So what is the enzyme used to break down the carbohydrate? Do you remember? Still remember how what is the enzyme used to break down the carbohydrates? Okay, so you can say we use enzyme amylase. Okay, amylase used to break down, and then after that chromatography, locating agent, measure the RF values, compare with what standards to identify. You need to mention the compare with standards to identify any standards. Okay, you don't need this one, you don't need to be precise because you can compare with any standards of amino acids. Okay, so we have discussed about this thing. Okay, broken down by hydrolysis, the process, the acid we use hydrochloric acid, dilute hydrochloric acid. Please don't say very concentrated hydrochloric acid because that's for protein. Okay, so you use dilute hydrochloric acid, you heat it, okay, and then you get the monomer. So you also use enzyme. Okay, you can say that you use enzyme amylase. Okay, this one you already learned in biology classes, I guess. So now we're going to discuss about chromatography, locating agent, measure the R. Okay, so what is the paper chromatography? Paper, yo, where's my pointer option? So paper chromatography is another way to separate substance from mixture. The mixture dissolved in a suitable solvent. Okay, you can use any solvent, you can use water, but actually there is not, the polarity of the water is not really good, alright? The suitability will be good, but the polarity, the scale of the polarity of water is not really good. Okay, so you can also use rubbing alcohol, for example, ethyl type or denatured alcohol, ethanol, for example, they have high polarity, so means that it's very good suitability. You also can use rubbing alcohol, isopropyl type. You also can use vinegar, but vinegar is the medium polarity. What means by polarity? Um, Polarity means the properties of her, what they call polarity. Polarity. Wait. Polarity. Okay, polarity. Polarity. So polarity is what do you call this thing? Refers to the way which atom bonds with each other. So the higher the higher the polarity, the higher the intermolecular attraction. But you no need to know this polarity yet. Okay, I just simply explain to you because you're going to be learning in university about this polarity things. But usually we use chromatography. The best of chromatography that we can use is the solvent that has high polarity. For example, isopropyl alcohol, denatured alcohol, ethyl type of alcohol. Usually we just use ethanol. Ethanol is just good. It has a high polarity and the sustainability is good. Right? So we use the chromatography paper, or yeah, we can also use what do you call it, filter paper. So you see the mixture of substance dissolved in the solvent, they travel at different speeds, so they separate because of the different solubility in the solvent and attraction of the paper they travel over. The more soluble substance in the solvent, the further it will travel. So the more attracted is to the paper, the less far it will travel. So that's why we need to have our solvent is very important, okay, because of this polarity things, because the attraction, okay, attract the intermolecular forces attract. Let's say uses of chromatography paper. So why you want to use this chromatography paper? To find out many substances are present in the mixture, to check the purity of the substance. Okay, another way to check the purity or impurity of the substance by boiling point. Okay, boiling and the melting point of the substance. Okay, for example, water has 100 degree of boiling point. So if you boil that substance, for example substance X, and the substance X show you 102 degrees Celsius, mean that substance is not water, because the boiling point for water is 100 degree Celsius. So you can check the purity of the substance by this chromatography, or you can check by boiling point and freezing point or melting point of the substance. Okay, so to identify the substance in the mixture by measuring how far they travel. Okay, so this is how you do. You need to explain the chromatography. So you need a solution, the solvent. Okay, you need to mention what solvent you use. If you don't have any idea, I want what solvent I need to use, teacher, just say ethanol. All right, don't say water, because like I say, water is not really good polarity. I mean the polarity of the water is not really good, so just use ethanol. Okay, ethanol. And please make sure the baseline mark in pencil. Okay, chromatography paper, and you need to mark in pencil. Why you cannot mark in pen? Because it might be dissolved in ethanol. So solvent, make sure the solvent is below the line of your baseline, alright? And then allow the water to rise up the paper. When it's near the top, remove the paper and dry it over. So you have the several spot, but usually amino acid colorless. Okay, so what happens if you have colorless substance of amino acid? So if the amino acid is colorless, you need to spray it with ninhydrin, or you say locating agent. Okay, ninhydrin is a stable one. Okay, one example of locating agent, ninhydrin. Usually we use ninhydrin. So after that, you can see the color. These 3 purple spots appear, so that the solution contains 3 amino acids. Okay, so please make sure you state that you also have what they call the thing, standards. Okay, what standards, teacher? Just measuring you had you measured you have standard to measure what kind of amino acids you have acid. Okay, so for example amino acid, this kind of amino acid has valine type, so the standard is valine type, so you can see the dot. Okay, for example you have to have standard. So for example, if I draw here, okay, this is your standard. Okay, you have one spot here, this spot is for valine, so it has same spot for valine, so the amino acid type is valine. Okay, so after that you need to do the RF value. What means RF value? Distance move by amino acid divided by distance move by solvent. This is distance moved by amino acid divided by solvent. So you get the RF value. You just mentioned you need to calculate RF value. Okay, understand? So that's all for 6 marks question. Like I said, 6 marks question, you need to be precise. If you can describe that thing, you describe that thing. How many mil of hydrochloric acid you use, what kind of acid you use, what is the process, what enzyme you use, and then how to do this chromatography. Okay, and then what type of solvent you use. Okay, be specific. Okay, that's all for now, guys. All right, thank you for listening. I hope you understand the topic, the how to answer this paper 6. Paper 4 question 6 marks. I'm sorry, paper 4 6 marks question. Bye.